I view common bean as a major societal crop because it provides both food and financial security to those with severely limited income options. Most consumers are familiar with pinto beans, kidney beans, black beans, and navy beans. All of these are common beans, a member of the species Phaseolus vulgaris. Snap beans or string beans are also members of the same species. Hi, I'm Phil McClean from North Dakota State University in Fargo, North Dakota. Along with Jeremy Schmutz and Scott Jackson, University of Georgia, I was a co-principal investigator on the first common bean genome sequencing project.
My name is Jeremy Schmutz, and I'm a faculty investigator at the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. And I'm also the leader of the plant program at the Joint Genome Institute in Berkeley, California.
From a health perspective, common bean is a major source of protein. For some countries like Rwanda, beans provide up to 30% of the daily protein requirement. It is also a rich source of iron and zinc. For growers in those regions, common beans is also a cash crop. The cash they generate from the sale of part of their production goes to support the purchase of other items needed for the family. Common bean is also a commodity crop, especially in the United States, Brazil, Mexico, and China. For smallholder and commercial farmers, diseases are the major production problem. Because of the major impact diseases have on production, the reference genome has been an important resource to discover resistance genes to the many pathogens.
Technology was actually quite a bit different than it is today. There was actually only about 15 genomes for plants that had ever been sequenced before. And we're talking about back in 2009, 2010. And that was because the genomes were really a lot of effort and work, and the technology was really just developing post-human genome to be able to sequence a whole genome of a plant or a person and put it back together again. It was incredibly expensive and time-consuming and involved lots of different players and researchers across many different labs to be able to sequence a genome.
I think it's also important to note is that back then, there was about $2 million of JGI money and $1 million from the USDA. So the original genome cost around $3 million. And I think the latest ones we're doing right now are about 20K. And they're much, much better.
Common bean was kind of an interesting project because at the time, genome sequencing of plants had really just started, and the JGI and the Department of Energy was sequencing several different plants, and we were working in tandem in collaboration with places like USDA or NSF funded researchers in order to be able to sequence these reference genomes. And so the agencies work together on different projects, and Common Bean is a great example of a project of the Department of Energy Joint Genome Institute working together with USDA funding researchers and NSF funding and researchers to be able to put together a public resource, a public reference genome for Phaseolus that anybody who wants to in the entire world can use for their research projects.
Recently, the reference genome has become important to discover the genes associated with resistance to heat stress, water stress, and low fertility.
It is amazing. Yeah, I mean, I think, Phil, from the perspective of where we've gotten to in plant genomics, like you're saying, well, it's nice now that we can have multiple different genomes of the different race classes so we can compare between them.
Beans are beans. Whatever diseases you find in pinto beans, you'll find similar diseases affecting snap beans or navy beans or other ones. So when you're trying to make discoveries, you're going to end up making discoveries across all the different marker classes of beans. It's not just specific to one particular marker class.
We have sequenced many more genomes of Phaseolus now, and the technology has continued to improve over the previous 12, 13 years now. We've gotten now long single molecule sequencing, where we're sequencing something like 30 to 50,000 base pairs at a time for a single sequencing read. And this is a comparison to the original Phaseolus, which was about 500 base pairs long. And so what that means is that when you're putting together this very large puzzle of a genome, we have many less pieces to work with now these days than we did with the original Phaseolus genome. And so that it's a order of about 100 times less number of pieces. And that enables us to really now produce these incredibly high quality contiguous versions of these Phaseolus and bean genomes.